Hello everyone, welcome back to our YouTube channel Agri Addict. I am your mentor Hariprasad SK, a PhD scholar from Indian Agriculture Research Institute, New Delhi. Today we are going to discuss a topic of biotechnology in which we will be discussing about recombinant DNA technology. So this recombinant DNA technology is very much important for all the upcoming ICR PG aspirants if you are going to write your exams in the subject of either plant science, and it is most important in case of the subject biotechnology. Okay, I hope like you people have prepared well for your 2024 exams. So just watch this particular video which might be very much useful if they are going to ask any questions related to the biotechnology. Okay, and we are going to start from the scratch and we will be discussing each and everything in detail about this recombinant DNA technology. So stick with me until the end of the video and take a full benefit out of it. So the content outline will be, we'll be discussing about this recombinant DNA technology, which is also called as genetic engineering, about its introduction, why and what is this technique, and then we'll be discussing about the brief history, like who developed it, and then we'll come up with the what exactly is the recombinant DNA technology. And next we'll take a method under the recombinant DNA technology, which is a gene cloning or a DNA cloning, and we'll discuss this in detail. And also we'll discuss the different types of approaches we have for the effective gene cloning and its complete procedures. So this is the outline. This is what we are going to discuss in the today's video. So if you like our content, please like, comment, share and subscribe our YouTube channel. It will definitely encourage us to provide more such quality content. And if you have any doubts regarding whatever content that I have dealt in the YouTube, you can comment on those videos. We'll be looking into the comments regularly and will be definitely answer all your queries. None of the answers will none of your comments will be left unanswered. Okay, so firstly let us start with the recombinant DNA technology. What is this genetic engineering? So to understand this genetic engineering, let us have this example. For example, if I am developing a variety, okay, I want to develop a variety which is resistance for a certain disease, which has a resistance for certain diseases or the pest. Let us take a disease now. Okay, so what I will do, I will look into the plants within the species or in the complete germplasm whether there is a resistant for the particular disease or not. So if I am not able to find the resistant lines or resistant plants which are actually resistant to the diseases of my interest, then I will be looking into a sub different species or a different organisms. By any case, if I find any gene that is able to produce the resistance to the my gene of like my disease of interest, then there is an opportunity that I can take out the gene from the that organisms and I can incorporate into the plant which I am developing, like the variety of a plant which I am developing. So this is just a transfer of a gene from a different source or from one organisms. Like the gene means it is just a short stretch. It is a short stretch of DNA sequence. So if I want to transfer the short stretch of DNA sequence or a gene from one organisms which is actually producing the resistance to a desirable organisms, so what I have to do, I have to take out the gene, extract the gene from the organism, and I can put it in the organisms of my interest, right? So this is a simple process for a conceptual thing. So this is what the rDNA technology explains. So this is a technology of the identification. First we want to identify our gene of interest or a DNA sequence of interest, and then we have to isolate that gene of interest or the DNA sequence. Then we can recombinant that DNA sequence into the suitable or desirable host. We'll take it as host or the organisms. So this is what our DNA technology explains, that is recombinant DNA technology, which is also called as genetic engineering. That means we are just trying to look into the useful DNA sequence or a gene. If it is not available in organisms like particular species, then we can look into the different organisms, and then we can take it out and put it in the desirable organisms to have a commercial benefit. So this is about the rDNA technology. Okay, next. So I'll just show you what I have said in the previous slide. So I told you that taking out the DNA from a certain species and incorporating it with the DNA of another species. So let us try to understand this in a other way. So I told you that segment of gene or a segment of DNA is being incorporated into a certain host. So this is also a technique under recombinant DNA technology, right? Similarly, if we have two separate or two different DNA sequence or two different genes from two different species, we can recombine them to form a recombinant DNA. This is also under recombinant DNA technology. This thing, it can be clearly showed in this picture. So you can see there are two different types of sequence. One DNA is from species 1 and other DNA is from species 2. So in the first step, what they have done is the cuts have been made by the use of certain enzymes which are called as endonuclease, restriction endonuclease, about which we will be discussing in the next video. Okay, just for now understand this, try to just know it. Okay, this is by the enzyme restriction endonuclease. So once the cuts are made, you can see there is a identical cuts at both the DNA since the same restriction enzyme is used. After the desirable cuts are made, the DNA from the other species is going to be attached with the DNA of this first species. That means you can clearly see there is a recombination that is being taking place. That means there are two different stretch of DNA, but a certain stretch is going to be added into one species, and certain stretch of the species one is added to the second species, and this is what ultimately we call it as recombinant DNA molecules. So this is about recombinant DNA technology. This is what it's been done. Okay, and for the first time, this recombinant DNA, under this recombinant DNA technology, it was for the first time showed by Paul Berg that uh, DNA sequence from the two different organisms can be clubbed and made into a recombinant DNA. So whatever I have showed you in this figure, the ultimate results where you are seeing the recombinant DNA molecules, that was for the first time showed by Paul Berg, and who is also called as father of genetic engineering. So what he has done is he has mixed the DNA sequence from the SV40 virus and the lambda phage, and he was able to recombine them, like these two viruses. Okay, so the ultimate recombinant had the DNA sequences from both SV40 virus and the lambda phage. So this was in the 1970s, and next it was in 1973, after the Polberg has showed that recombinant DNA molecules can be produced by using two different DNA sequence from the different organisms. It was in 1973 there were two different scientists that by the name Herbert Boyer and Stanley Cohen. So these two scientists, what they have done is they have showed that, or they have developed this rDNA technology, in which they showed the recombinant DNA, which consists of DNA from two different organisms. This recombinant DNA can be transferred into a foreign cells. That means it can be transferred into a living cell. If the living cell, for example the bacteria, if we take an example of the bacteria, the recombinant DNA, which is a mixture of the two different DNA sequence from the different organisms, that can be incorporated into a living host, and then this recombinant DNA can be replicated or created into multiple copies. And in the other words, for a simple understanding, for example, assume that I have a single copy of a certain stretch of DNA. Okay, for my area of interest is I want to do some research on this single stretch of DNA to know its gene, to know its complete sequence and also to know its function. But the problem is with a single copy of the gene, I may not able to do my studies, right? But I want, for that sake, what I want to do, I have to create the multiple copies, or I should have a multiple copies of this single copy of gene for the clear studies, right? So how can I multiply my single stretch of nucleotide or a single stretch of DNA sequence? So I can do it by using this recombinant DNA technology, within which there is a certain method called as DNA cloning, or It is also called as gene cloning, so which employs the method of this recombinant DNA technology, by which I can produce the multiple copies of a gene of interest. So what exactly the gene cloning or the DNA cloning is? The gene cloning or DNA cloning is nothing but the multiplication of a certain sequence of DNA or any gene of interest. So that would be a meaning of gene or gene cloning. So before I talk about gene or DNA cloning, let us see about the applications of this rDNA technology, why it is very much important in these days. The first thing is in the medicinal fields. For example, if we know about the diabetic patients, there is one type of diabetes in which the insulin production is stopped. That means the patient should be inserted with or given with the insulin externally. That means we want this insulin in a ample amount, that so that we can use it as a drug. So how the insulin is produced? Obviously the insulin is a protein, so which is produced by the gene, right? So but if we want to commercially exploit this insulin and market it and provide it as a drug, so we want it in a hundreds and millions of copies, right? Since like there are a lot of diabetic patients, but every time we cannot extract the protein directly from the humans and inject it to the other humans, so that is not possible in the real case. So for that case, what we can do is we can extract the gene that is actually responsible for the production of this insulin, and then we will transfer this gene into a suitable host which will allow this gene to independently replicate. And if this is a gene stretch of DNA and this is a host, like let us consider it is E. coli, okay, and we'll somehow transfer this gene of interest to this E. coli. Within E. coli, nuclear genome is also present in the nucleoid. So if in the through the binary fission, so the multiple copies of the bacteria is produced in very short period of time. So after the sufficient number of bacterial cells are produced, obviously like if the gene of interest is incorporated inside and it is allowing the replicate, there will be multiple copies of my gene of interest or the proteins that I want, so that I can extract at the end of the reaction and I can purify it and make it as a drug, right? So this is how it is being used in the production of drugs. But this procedure is not as such simple. It also required the aid of the vectors for the effective multiplication that we'll be discussing in the upcoming slides. Okay, next, similarly in case of vaccines, if we are wanting to produce some vaccines, so vaccines how they will work? So what we are doing, like if there is some certain disease that is because of and there is certain gene which is responsible for the production of certain disease, then what we can do is we can take out the DNA or the gene of or this gene, a part of this gene, and we incorporate it into the host cell in which we want to make it as a resistant one. This is how the vaccines work, right? So this is how we can use this rDNA technology. We just are wanting to isolate that gene which is actually responsible for the disease, and then we incorporated a part of that gene into the host which will provide the resistance to that particular disease. And next, there is a gene therapy. So in case of several disorders, the functional gene product is missing or the gene is non-functional. In that case, there is always a possibility that if that gene is available externally or can be supplied externally to that to the organism or to the humans, if there is a genetic disorders in the humans, we can correct or treat the disorder. In that case, this gene in the gene therapy, so we can use this recombinant DNA technology. Next, for the diagnostics. So it means to develop certain diagnostics test for certain diseases, first step would be knowing the sequence of that disease, like knowing the sequence of a gene that is producing the disorder. Knowing that producing the multiple copies, we can study that gene sequence and we can prepare the effective test which will actually detect these sequence. Whenever these sequences are detected, that means the disorder or the disease is present. So that is how we can clarify, and this is used in the diagnostics, and this is all in the medicinal field. Next, in the agriculture field, so we have a very common example which is bt cotton. So what is bt cotton? In the bt cotton, there are certain genes called as cry genes which are being incorporated from the which organisms, it from the bacteria to the cotton plant crop, right? So because of which these genes, cry genes, are resistant to the pest, provide the pest resistance in case of cotton. This has been done. So this is under genetically modified organism, and this is how the recombinant DNA technology is helpful in case of agricultural field. Next, similarly, if you want to incorporate any disease resistance, or if you want to enhance the milk production or the meat production, then we can take out the desirable genes, look into the desirable genes in any organisms, and then we can incorporate into the suitable or the desirable animal. Okay, so this is in case of animal husbandry. Next, in case of industry, see, we have several lot of enzymes which are very commercial, which are commercially important. For example, in case of textile industries, in case of food industries, we need the ample amount of, like, we need large amount of enzymes that will be regularly used in case of this industry. So how to produce these enzymes in a very large scale? To do it, we can employ this rDNA technology method, right? And next we have this bioremediation. See, bioremediation is nothing but the use of genetically engineered microbes to digest the pollutants, the oil spills, or any other harmful chemicals in the nature. So this is how the bioremediation is utilizing or exploiting this rDNA technology. Next, we have this biomining. So we already know what is mining, right? Mining is just extraction of the metals from the ores. But what is biomining? The mining which is done with the genetically engineered microbes for the extraction of the heavy metals from the ores, that would be the biomining. So these are some industrial applications. Next, so this rDNA technology is also having a scope in the research field also. As I told you, like, if I want to just study a certain sequence of DNA, if I want to do the gene function analysis, or if I want to discover a drug, or if I want to develop a genetically modified model organisms, in all these cases we can employ this rDNA technology. So these are certain very important applications of rDNA technology. So if anything is asked in the exams related to this application, I hope like you will be able to answer it. Okay. Next, coming back to the DNA cloning, I told you what is DNA cloning. DNA cloning is nothing but just the multiplication of desired DNA sequence or a target gene. That is what DNA or gene cloning means. So how the gene cloning or DNA cloning is done? The gene cloning or DNA cloning is done by two different approaches mainly. So what are the two different approaches? It is, one is cell mediated approach and second one is cell free approach. So the name, I guess, like, they are also already telling their meaning. Cell mediated approach means it requires a host and other cells like vectors for the multiplication of gene of interest. But in case of cell free technique, it doesn't require any host or the vector for their multiplication. Directly, if we have certain gene of interest, then by directly using certain enzymes, we can produce the multiple copy of this gene. So what is the example for this cell free technique? It is the PCR. I hope like many of you already know this term PCR, right? But we'll try to discuss in bit greater detail in the upcoming slides. First, we'll go for cell-based cloning. See, what happens in the cell-based cloning? This is a donor DNA. From the donor DNA, this is a desired piece of DNA. That means gene of interest, which I am specifically interested in. First, what I have to do, I have to use the restriction endonuclease and cut this specific part. After cutting this specific part, I have to introduce that desired DNA piece into a plasmid, so which is also called as vector is nothing but a vehicle which is used to take the desired gene and transfer it into the suitable host, and we will use the ligase to join this and form the recombinant DNA, which I explained in the very first slide. And after that, I introduced or transferred this recombinant DNA into the host. Here you can see that the complete plasmid, which is the recombinant DNA, now has been introduced into the host. And after the introduction, after the successful multiplication, as you can see, there are the multiple copies of my gene of interest along with the plasmid, which I can later isolate and extract them. Okay, this is about the cell based cloning. The simple words, if there is a gene of interest, what I have to do is I have to club it with a suitable vector, and using this vector, which is a vehicle, it will take this naked DNA to a host. Why the host is required? The host is required to multiply my gene of interest. So you might be getting at least one or two questions here. First is, why the vector is required? Why can't we directly transfer the gene of interest into the host and try to multiply our gene of interest? And the second thing, why we want to transfer my gene of into the host, why can't we do it without the host? The answer to your question is, first thing, the gene of interest which we are taking is should be replicated. That means multiplied means it should be replicated. But replication process requires what? It requires the complete replication machineries. So that includes the enzymes, and also most importantly it requires the origin of replication in case of prokaryotes. That means a replication sequence that will from where the replication starts. But any such of DNA, all such of DNA will not be having this origin of replication sequence, and the enzymes are not at present, so it cannot independently replicate. But this vector, the vector is the one which will be having the ability to replicate independently. That means they have their own origin of replication. So what type of vectors will be having the origin of replication? See, again the vector is nothing new, it is again the DNA sequence only. But what type of DNA sequence will be having this ability? They are plasmids. The circular genomes which are called as plasmids, which are present in the multiple copies in the prokaryotic cells like E. coli. So multiple copies of these plasmids is present along with the nucleoid in which the nuclear genome is present. This plasmid has an inherent ability to independently replicate over the generations in case of E. coli. So this particular plasmid can be exploited and used as a vector, and we can incorporate our gene of interest within the vector, and this vector will go into the host, and within the host this bacterial cell won't disturb its replication. It can independently replicate by itself and produce the multiple copies, and after certain copies are produced, after required number of copies are produced, we can take out the gene of interest or the protein, and we can purify it and use it commercially or for whatever purpose we wanted to do it. So this is about the cell based DNA cloning. I hope this is very much clear to all of you. Next, it would be cell free cloning. See, in the cell free cloning, I told you there is no usage of either the host or the vector. Then how does target DNA or the gene of interest is being replicated? It is by employing a method called as PCR. So what exactly is done in the PCR? In the PCR, what we are trying to do is, so this is our gene of interest. So in the first step, this is a three-step process or the three-stage process. Just remember, the PCR is a three-stage process in which in the very first step, which is called as denaturation, we are separating the two different strands. Like the double-stranded DNA is converted into two single strands, as you can clearly see by the breakage of the hydrogen bonds. How the breakage of hydrogen bonds takes place? It only occurs at very high temperature. The temperature should be 90 to 95 degrees Celsius, which is given for 15 seconds. That will break the hydrogen bonds. So the denaturation temperature is 90 to 95. So once it has been denatured, we have to synthesize the artificial primers, like synthetic primers has been designed, which will be complementary to the ends of this target DNA on the both the strands, and these primers will be attached to these two single strands at a temperature of 52 to 55 degree Celsius, and this process is called as annealing. And after this process, there is a last step which is extinction. In the extinction step, we can use the suitable polymerase enzyme which has the ability to elongate, the DNA strand. That means addition of nucleotide sequence. So we can use the DNA polymerase, by which, by since the primers are already added, it has a 3 prime free 3 prime OH group, and the polymerase enzyme can elongate this stretch of DNA and complete the copies. And if this particular process is repeated for the multiple cycles, we get the ample amount of gene of interest. But there is a problem here. So the DNA polymerase which we are actually using, which are usually used for the replication process, cannot be used in this case. Why? Because the temperature is very high. In the DNA polymerase will be degraded, it won't be active at this temperature. Even like if we add the DNA polymerase at the extinction step, even it can extend the DNA, like extend the DNA stretch in the next cycle when it is again going for the denaturation, which is at very high temperature. This DNA polymerase will be degraded, right? So at every cycle, at the extinction step, we have to add a new DNA polymerase enzyme. That is quite cumbersome, right? So to for this, there is a best alternative method that was established. by the use of a certain enzyme which is actually stable at a very high temperature. So what is the name of the enzymes? I guess like you might be already knowing this enzyme. So the enzyme is called as Taq polymerase. So this Taq polymerase is a enzyme which is extracted from a bacteria which is Thermus aquaticus. You have to remember this, and this is stable at very high temperature. This Taq polymerase is usually used in case of polymerase chain reaction. Okay, so the PCR. So what you have to know about the PCR? First of all, we'll talk about the requirements and then we'll talk about the disadvantages. See, if I have a stretch of DNA and I have to go for PCR, so then what are the requirements? First thing is I have to know the sequence, the complete sequence of my gene of interest. Only then I'll be able to produce the synthetic primers. At least we have to know the ends of this gene of interest. Only then the synthetic primers, which are RNA primers, synthetic RNA primers, which are complementary to the both the strands, can be synthesized and they can be attached. So this is the basic requirement for the PCR. Next, so then what are the disadvantages of this PCR? So if you clearly look into the Taq polymerase enzyme, similar to that of DNA polymerase, it has 5' to 3' polymerase activity. This is how it is able to elongate the chain, and it also have the 5' to 3' exonuclease activity, which will remove the primers, right? But it will remove the primer removal, but it doesn't have a 3' to 5' exonuclease activity. So what is the function of this activity? It is required for the proofreading. That means like it will check for any errors during the replication, but this activity is not present in the Taq polymerase. So there will be a more number of errors than normal that will be incorporated when the PCR is being used. So this is the one important disadvantage of the PCR. So compensate this, there are now new sets of enzymes that have been used based on the requirement. If they want to like create the multiple copies of a genome which are very much accurate, in which there should be no errors or very minimal amount of errors, they are not using the Taq polymerase enzyme now. So they are using other enzymes, advanced enzymes, which are actually having this 3' to 5' exonuclease activity. Okay, so this is about the disadvantages of the PCR. So that's it for today's video. In the upcoming next video, I'll be discussing about the enzymes for the DNA manipulation, which will include the restriction endonuclease, which I told you in the previous 2 or 3 previous slides, right? Like while the cutting of DNA, we require this enzymes. So we'll discuss in detail about what are the functions and how it is going to act, and what are the different categories and classes. Everything will in detail we will be discussing in the next class. That will be important for your exam point of view. So if you like our content, please like, comment and share our subscribe our YouTube channel. And also if you are looking for the complete mock test bundle for your upcoming ICR PG exam, which is going to be held in another 2 days, you can purchase our mock test bundle, which includes the topic wise mock test, the subject wise mock test and the whole syllabus mock test, just for 149 rupees. Okay, the links for the purchase will be available in the description box. Either you can go for the application directly, or you can visit our website www.agriaddict.in and you can purchase the mock test. And if you are wanting to and if you are looking for a free mock test for your subjects, then you can visit our website, in which for all the subjects we have 2 sets of free mock tests available that you can attempt. So attending this mock test will definitely help you in evaluating yourself in a better way and giving your best shot in the exams. Just try this mock test, and if you have any queries, you can always comment on this video. I will be happy to answer. Thank you.